Dr. Branden Moriarity is a hematologist in the pediatric division at the University of Minnesota. He has done landmark efforts, especially in areas of osteosarcoma. Been written up nationally and internationally on his work in that area. Dr. Eva Galanis is head of molecular biology at the proper term area at the Mayo Clinic. Is going to talk about your best friend, which is viruses.
So yeah, just want to thank everyone for coming today and having the opportunity to show you guys kind of what we've been doing at the University of Minnesota for a couple different kinds of sarcomas and trying to identify new treatments for those sarcomas. So the title of the talk is Identification and Testing of Novel Actionable Targets to Treat both osteosarcoma and a tumor type you may not have heard much about called malignant peripheral nerve sheath tumors. So first I'll just give you a little bit of background on these two different tumor types. So my main work focuses on osteosarcoma. And osteosarcoma is a cancer that develops from bone cells, so the cells of your body that actually produce your skeletal system. Here's just an x-ray here showing that there is solid bone in these tumors, and also just a gross anatomy. So this happens usually in adolescent children, largely in kind of the long bones or the rapidly developing parts of the skeleton system. And so this is a fairly rare disease, about 900 cases annually in the United States, but it's also a very deadly disease as well. In fact, until the advent of chemotherapy in the 1960s, it was a pretty dismal survival rate of less than 20%. And so now with chemotherapy, we can cure about 70% of patients with osteosarcoma. However, if they have metastatic disease when they're diagnosed, that is, the tumor cells have spread to other parts of the body, that rate of survival then drops to about 30%. And so the real problem here is that we haven't developed any new therapeutics for this cancer type in the last 4 decades. So we really need to put a lot of effort into identifying new targets to treat osteosarcoma with better drugs and other therapies. So the other type of cancer that we've spent a lot of time working on, as I mentioned, is this MPNST, or malignant peripheral nerve sheath tumors. So these are tumors that actually develop from the Schwann cells that are associated with nerves throughout the human body. So about 10% of the patients with this genetic disease called neurofibromatosis type 1, they get this disease very commonly, but approximately 50% of the cases occur from sporadic disease as well. And it's also a very poor prognosis, with about 25% of the patients surviving if, again, they have metastatic disease. It's treated analogously as osteosarcoma is, where they simply remove the tumor and then treat with non-specific chemotherapy. And again, there have been no new therapies in the last several decades for this disease. And just to give you an idea of why it's been so difficult to develop new therapeutics for these two cancer types. And so what I'm showing you here is what your normal chromosomes in your cells should look like. You have 2 copies of every chromosome in general. Now in these tumor types, when we look at their chromosomes, you can see that they've massively disorganized. And essentially what's happened is they've had amplification of bad genes and loss of lots of good genes. And it's this complexity of these tumor types that has made it really hard to develop new therapeutics for, because we don't know what's driving these tumors. We don't know the cause of them. And so that's kind of where we come in at the University of Minnesota. I started this work in David Largaspada's lab when I was a graduate student, and also with a colleague that was a graduate student in his lab, Dr. Eric Barman. And so 7 years ago, we set out with a mission to identify the causes of osteosarcoma and malignant peripheral nerve sheath tumors, because if we can identify these genes that are causing these tumors, we can develop new, more effective, and safer therapies to treat these sarcoma patients effectively. And so the way that we set out to do this was using a new technology that David had developed here at the University of Minnesota, and now has been adapted worldwide to studying different types of cancers. So essentially what we do is we use mouse models, and we developed a system using what we call jumping DNA elements. So these are small pieces of DNA that in the mouse cells, they randomly jump around, and they can either activate bad genes or turn off good genes to induce cancer in the mouse. And so the really critical part about this system is we can identify the genes that are causing these tumors no matter how complex they are. So with the human tumors, we can't figure out which genes are causing them. But in the mouse, we can directly identify using this novel technology that we developed at the U of M. And so essentially, we developed many tumors of the same type in many, many mice. So I bred just hundreds of mice, and then we ate some for tumors. And they look at all the tumors, and what we see is that certain genes in the genome are targeted by these jumping genes more often than we'd expect by chance. And these are the genes that then are involved with the tumor development of that cancer type. And then we can go on and study them further. Now, knowing which genes they are, we can look at the human tumors and look just at those genes and figure out if they also cause that tumor in humans. And so the results of my project for osteosarcoma are shown here. Essentially, these are all the genes that we identified, and the larger the gene, that means that it affects more of the different tumors, so it's more prevalent. And so essentially, we generated about 120 osteosarcomas in mice. We identified all of these genes, not only for primary tumor development, but also for metastasis. So now we have new targets that we can work on to develop therapies specifically stopping metastasis of osteosarcoma. And we also identified genes that were somewhat known to be involved in this cancer, actually validating that we are identifying these genes specific to osteosarcoma. And when you look at the chromosomes of these tumors, they have perfect 2 copies of every chromosome because the tumors were caused by the jumping genes. So the genes were identifiable for the first time ever of osteosarcoma. And so now we have this list of genes that cause osteosarcoma. So what can we do with this to actually lead to the development of a new therapeutic for osteosarcoma? So the first thing that we do is we take these genes and we look at all of the published data on any genes by any scientist in the whole world, and we identify that these genes are working in certain pathways. It's kind of like an instruction pathway of the cell, and we can specifically make drugs to target those pathways. And this is more effective sometimes than targeting single genes. And so the other really important thing that we did is we looked at the genetics of many, many, many human osteosarcomas. And what we found is that the genes we identified in the mouse are actually also driving the human tumors as well. And so what we predicted we would find, we actually did. And then the last step is really where we're putting a lot of effort in the lab right now, and that is now we have these genes, now we need to develop new therapeutics or use existing therapeutics that are made for those targets and then test them in osteosarcoma cell lines and mouse models in the lab so that we can then translate these bench to bedside into the clinic. And so that's where a lot of the work is focused now in my lab. And so out of all of this work over the last 7 years, we identified specifically one gene that we think is critical to many osteosarcomas. And this gene's name is called semaphore D. And we think that this is a novel, actionable therapeutic target to treat osteosarcoma patients. And so this slide kind of summarizes years of work, but the basic punchlines here are that in our mouse models, we see that when the jumping genes land near semaphore D, they overexpress a lot of this protein, and that can cause the tumor. And when we look at human tumors, we actually see that many of them express this gene semaphore D. In fact, over 50% of all osteosarcomas express high levels of this, and therefore we could potentially treat all of those patients using a drug that can inhibit this particular target. And so in the lab, when we synthetically turn on this gene a lot, we get really large tumors. And when we repress this gene, we also see that there's a large reduction in its ability to grow and can actually lead to the death of the cancer cells. So this is all in the laboratory setting, though, so we need to now somehow translate this to the clinic. And so we were really lucky that we found this in osteosarcoma, because it turns out that this gene is also causing other tumor types, but was never known to be causing osteosarcoma. So in fact, there are already drug companies that have developed drugs that can inhibit semaphore D. And so as you can kind of see here, you can imagine if this is a cell, semaphore D normally tells that cell to do functions such as like grow really rapidly, to migrate to other locations, and essentially just survive. And if we use this, this drug called an antibody to semaphore D, it actually blocks that protein and no longer signals to the cancer cell. And so the cancer cell loses these abilities to grow uncontrolled and also to migrate to other places. And so we're really excited about this, and we've partnered with a company that makes this therapeutic, and we're now testing it in the laboratory, and preliminary results on osteosarcoma cells show that indeed it does make them less aggressive, they grow slower, and sometimes they can even be killed by this drug. So this is the really exciting part about this. So not only can we kill or slow down osteosarcoma cell lines with this drug also has the ability to alert your immune system that you actually have a tumor. And it tells your immune system to go traffic to this tumor and start killing tumor cells. And so this therapeutic will not only stop the cancer from growing, but it will also activate your immune system and get your immune system to travel to the tumor and kill the tumor. And so we think this dual effect of this drug could actually produce durable cures for osteosarcoma patients. And so we were interested, since this was never known in osteosarcoma, could it be that other sarcomas are also being driven by this gene, semaphore D? And so we looked at other sarcomas such as Ewing's and also rhabdomyosarcoma. And here you can see, this is just kind of like how much of this is in the tumors. And some of these other sarcomas also have very high levels of semaphore D. And so we're right now testing this drug in rhabdomyosarcoma, Ewing's sarcoma, and fibromyosarcoma because we think it could be therapeutically relevant to those cancers as well. And so I'm kind of just going to end here with some future directions of our goals in the lab and also translating our results from bench to bedside. So we're continuing to assess the therapeutic efficacy of this drug against semaphore D and osteosarcoma and many other sarcoma cell lines. And we're starting now to treat mice that have osteosarcoma with this drug so that we can not only study its ability to kill the osteosarcoma cells, but alert the mouse's immune system to traffic to the tumors and eradicate those tumors in the mouse. And lastly, we're working with Branden Moriarity at the University of Minnesota and the Children's Oncology Group to start a phase 1 clinical trial to actually treat osteosarcoma patients at the University of Minnesota with this new antibody therapeutic. And so that's in the works, and we hope to have that going in the next year or so. And so with that, I just want to end with some acknowledgements. So all the hardworking people in our lab, including Natalie Wolf, who's here today, and a sarcoma survivor. And then all of our wonderful collaborators, and David Largaspada specifically, for helping us to come to fruition. And then many funding sources, the SOBIEX, AACR, and of course Branden Sarcoma. We really appreciate your support. And with that, I'll take any questions that you guys have.
Is this the only place they're doing that trial? Will it be at Mayo Clinic? Or?
We will be the first people in the world to have a clinical trial for osteosarcoma and also sarcomas with this drug.
I know it's predominantly in children or young adults, but it can affect older people too, is that right?
That's right. So osteosarcoma has the peak incidence in children and adolescents, and then it's a bimodal peak, so there's another peak actually later in life in the 60s, and it can be associated with radiation therapy or large exposure to X-rays as well can induce osteosarcoma. Yeah, so right now we're finishing
up the question.
Yeah, so she was wondering about the timeline for the clinical trial. So right now we're finishing the preclinical studies in my laboratory, and then we'll use that data to write the letter of intent to initiate the clinical trial. So when it will actually start, I'm not sure, but the letters of intent to actually do it will be submitted in a month time frame. All right, thanks a lot.
Good afternoon. It's a true pleasure and honor being here. As Peter said, not only I have a laboratory, I design and conduct clinical trials, but I also have a clinical practice. And that puts all the effort here in a whole different perspective, because we're not talking about theoretical things. We're talking about patients I see every day. And I know that our patients don't have 5, 10, 15 years to wait. We need answers now. And along those lines, I'm very honored to be here today because I think the theme of the afternoon is what are some of the efforts that are happening here in our state, in Minnesota, that they bridge the laboratory with the clinic and bring to our patients treatments never tried in sarcoma patients before that perhaps can completely change the landscape of how they treat these tumors. So I'll take the next 10 minutes or so to talk to you about my passion, which is using viruses to treat cancer. Why viruses? So viruses are one of the most destructive forces in nature. And what they know to do very well is to destroy tissue. So this is what we thought. Let's just take this destructive power and harness it and make it cancer specific. So the way that a Time article about this work described it, take these really bad actors and make them soldiers to fight cancer. Now, as you possibly remember for your biology class, viruses can be divided into two big categories depending on the type of genetic material they carry. And I want to show to you viruses we and others have actually used in the clinic to treat cancer, both DNA viruses. For example, you can see here adenovirus, which is the common cold virus. Herpes simplex virus, that's the virus that causes the cold sores, and I will come back to that. Vaccinia, which is the virus that has helped eradicate another lethal disease, smallpox, and as well as RNA virus. And of these RNA viruses, these two viruses, the measles virus and the vesicular stomatitis virus, this bullet-shaped virus, these are viruses that our group at the Mayo Clinic first took to clinical trials. And we will talk a little bit also about the results we are seeing in other tumor types that are truly very promising. The other thing I want to convey is that this is not scientific fiction anymore. I mean, it sounded this way 20 years ago when I started working in that field. But in 2015, for the first time in the US history, the Food and Drug Administration approved a replicating virus for treatment of cancer, and that's the herpes simplex virus that improved outcome in patients with melanoma. And we hope there are going to be many more tumors to come for which we will have active replicating viruses to help our patients, and we hope that sarcomas will be one of these tumors soon to follow. So I'm sure people are wondering, why measles? Our interest in that virus is based on reports back in the 70s where children in developing countries who happen to have different tumors, like this boy with a big lymphoma, you can see he cannot open his eyes, then got infected with measles virus and the tumor disappeared. So that made us think, well, there must be something about this virus' ability to kill cancer cells. Now, we would not want to use the wild-type virus. The reason that we vaccinate our children against measles is because the wild-type virus can occasionally cause serious disease. On the other hand, the vaccine strains have an excellent safety records. Millions of people have been vaccinated. We give the measles vaccine to the newborns. So we thought, well, let's just start trying the vaccine strains in the laboratory and see what happens. See if we can both maintain the anti-tumor effect and the excellent safety. And we're actually very pleasantly surprised to find out that the vaccine strains not only are as effective as the wild-type virus, but actually they're even more effective. Because the way that we grow the vaccine strains in the laboratory, they enter now cells via proteins, we call them receptors, that are overexpressed in cancer cells. So that's why the vaccine strain of the virus kills tumor cells much more efficiently than normal cells. And you would argue, well, what is this kind of protein, the receptor, that allows the measles vaccine strain to enter the tumor cells? These are two proteins called CD46 and Nectin-4, and what these two proteins do, normally, they protect the tumor cells from the immune system. So the virus can use these very receptors as the Trojan horse to enter the tumor cells and then destroy them. And what is actually quite interesting is the way that the infected cells die. When a cell is infected by the virus, then it fuses with neighboring cancer cells. So what we have shown in the laboratory is that for every cell we infect, another 40 to 100 cells come together, melt, fuse, and then they commit suicide collectively, what we call apoptosis. So, and this is what we call a bystander effect. You kill many more cancer cells that you infect. Going back to the discussion that Branden led a few minutes ago, you all know how it goes. On one hand, we have a lot of clinical need. And every one of you who has ever dealt with cancer or took care of someone with cancer knows that. There is so much need in the clinic every day. And on the other hand, we have promising preclinical work like what we heard from Dr. Moriarty, the work that we others are doing, and bridging the two. It should have been straightforward, but it's not. It requires a lot of regulatory approvals, it requires time, it requires resources, because now your laboratory reagent becomes a drug. It needs to be safe for the patients, first to not harm, but in addition, it needs to be, to meet very strict standards, especially when this drug is a live organism, an agent that replicates. One thing I'm particularly proud about is the fact that we have been able to move this process fast, and from the first proof of principle experiments in different tumor types to our first clinical trials, it was an average of 2 to 3 years. And that comes back to the dedication of a team of people who want to find a solution for patients with cancer soon. So we have studies for ovarian cancer, glioma, multiple myeloma, head and neck cancer, mesothelioma, which is a tumor of the chest wall cavity, and also a study in sarcoma now open this year, and we're gonna talk about that later. As I discussed, when you try to move a laboratory drug to the clinic, there are two things we need to address: production of the drug and safety. At the Mayo Clinic, we actually have a Food and Drug Administration approved facility that produces the viruses. This is a snapshot of how inside the facility looks like. You see the members of the facility wearing this astronaut type of suits. As the director of this facility nicely said, the dirtiest thing in there is the humans. So we need to completely make sure that our product, what we give to our patients, is sterile and meets the highest standards for industry. And then, of course, we need to make sure that it is safe, and we do that by testing it in appropriate medical models. Of course, the price we have to pay for novelty is the extensive toxicology testing. That was the very first time that a replicating virus, such as measles virus, would be used in humans. So the FDA made us test it in 3 different models, including primates. And everything was safe and good. But then I can tell you that there is nothing that prepares you about the anticipation and anxiety when the first human is treated with a drug never been tried before. And this is our very first patient, and I have to say, in this journey, our patients are my heroes. They are the ones who inspire us, and also they are the ones who encourage us. So in that room, with full of a bunch of people who are so anxious about this replicating virus being given to human, our patient, Dixie, was the one who was smiling and was the calmest of all. And interestingly enough, as sometimes happens, the very first patient treated with the virus also had a wonderful clinical response. So I would meet with her and I would try to rationalize. I would say, oh, well, you know, yes, your tumor is responding, the tumor marker is coming down, but I don't know if it is the virus. So I think she had it with me. So one day she looks at me and says, you know, doctor, I don't know what they teach you in the medical school, but for my disease, I know only when a treatment works, my tumor responds. So again, a source of encouragement for us, and the reason we're doing all of that. And since then, since Dixie was treated, which was about 10 years ago, we have given measles virus through different routes of administration. In the tumor, in the resection cavity when we remove brain tumors, intravenously, in the peritoneal cavity, and in the chest cavity, and has been extremely safe in all our patients. Now, then the question is, does it work? We believe actually that there is significant activity we have observed so far, and I want to share with you some of the reasons we think that. In addition to this first patient, we had a number of patients where we observed response in the tumor per se. This is another ovarian cancer patient, and her abdomen at the time of surgery had this kind of appearance, all these blue tumor cells in the abdomen. So the surgeon tried to remove as much as they could, but you know ovarian cancer actually spreads in the peritoneal cavity, so at the end of the procedure there was a lot of tumor left. 6 months later, after 6 doses of treatment, when we went in for what we call a second look surgery, that what it was left, fibrous tissue. And this patient remained disease-free 3.5 years, and when she relapsed, she never relapsed in the abdomen, she relapsed in her chest. And these are the data from all the patients with ovarian cancer we have treated so far. It's about 60 patients now. And what you can see here is that despite the fact that these patients have had lots of chemotherapy regimen, the average patient in our study have seen 3 or 4 prior chemotherapies, where we expected them to live up to 6 months. You can see that patients in the study lived an average of 28 months. So we're very encouraged with that, and we're moving that to the next step, what we call a randomized phase 2 trial, where we compare this approach to physicians' chemotherapy of choice. Because one important thing, in addition to this promise we saw here in survival, was the safety. Patient had absolutely no toxicity, no relationship to the toxicity that patients experience with chemotherapy. And this is an example from a different tumor type, multiple myeloma, which is a disease that affects the bone marrow. And that was one of our patients in her 40s who had failed everything that there is out there for her disease, failed chemotherapy, multiple lines of chemotherapy, failed stem cell transplantation twice, and the cancer came back. You see here this cancer deposit in her forehead, as well as in her bone marrow. She was treated in May of 2013. By the end of the year, this had disappeared, the bone marrow had cleared, and this patient is still disease-free 2.5 years later. So there is something very potent about this technology that we want to understand for 2 reasons. Not any different than other drugs. Not all drugs help all patients. We need to be smarter about how we select the right patient for this approach. So we have done, and I'm sorry that does not show very well, but we have done very extensive work to try and understand that. This is an example. So this is a patient, a brain tumor from one of our patients. We put the catheter in the tumor, we inject the virus. 5 days later, we remove the tumor and the catheter, so we can characterize how far from this catheter the virus has spread and if the virus can replicate. And you can see here a picture from this tumor. Brown means viral proteins, and you can see the virus replicates, that's measles virus, replicating very well in this brain tumor. But also, we went a step further and we performed analysis of the genome of the tumor, of this tumor, prior to the patient being treated. And data that we have now accumulated from this and other patient suggest that patients who have a particular gene signature that blocks their response to the virus are the ones that allow the virus to replicate better. So what we really want to accomplish moving forward is come up with a gene signature that will allow us to select patients that will have the most benefit from this treatment. So how about sarcoma? So I left the best for last. Work done in my laboratory by Evidio Domingo and other people in other labs in molecular medicine indicate activity of these viruses against sarcoma lines. And we tried it against osteosarcomas, soft tissue sarcomas. Just to explain what we're doing here, we actually used a virus a measles virus that encodes a gene that when it infects cells and replicates, produces a green protein. So green cells are infected cells. And similar here, soft tissue sarcoma cells using the vesicular stomatitis virus, the bullet-shaped virus I told you about. And these are the malignant peripheral nerve sheath tumors that Dr. Moriarty also talked about. And you can see here treating these tumors with increasing doses of virus. Red here means, violet I should say, means surviving cells. And you can see as you increase the dose of the virus, you completely eradicate these monolayer cultures. And this is not just something we see in the cell culture, that's something also we have been able to see in animal models. This is work done using patient-derived lines from malignant peripheral nerve sheath tumors and then establishing animal models. And you can see here that treatment with the virus suppresses the growth. Here are the treated tumors. You can see, for example, from this line, there is essentially no growth at all. And then, when you test it in animals with the tumors, the control animals invariably die fast. This is days after treatment and percentage of animals surviving, while treated animals live longer, or actually, in this model, 60% of the animals are cured. So the basis of this data, FDA gave us the approval, and actually the Department of Defense funded a clinical trial we'll be activating in 2016 for patients with these tumors. But also, we saw similar activity in patients with osteosarcomas, this time using measles virus or vesicular stomatitis virus. And you can see again the control animals dying fast. And this is, we used orthotopic models, so models where the sarcoma grows in the bone, same way that it does in patients, while when you treat with the virus, you significantly prolong the survival.
We can both get you behind the table down here in area. We'll take any particular questions. We had Dr. Closey and Dr. Okuno here last year at this area, and we kind of cautioned you again talking about here about your personal situation. We're looking at what you think may have to be germane to other people as well as yourself, and that's good. And I think Dr. Okuno mentioned it felt like it was a public radio audience after we finished, so we'll see what you come up with. Michelle.
I'm just curious about Dr. Moriarty, if you can tell us a little bit about your professional path and where you started and how you ended up taking particular interest in osteosarcoma.
Sure, yeah. So I did my undergraduate work in Northfield, Minnesota at St. Olaf College. And then I came to the University of Minnesota for my graduate work, working in David Larry-Spada's lab. And at that time, he was just starting to develop this new technology I talked about where you could identify genes. And he had an urge to work on pediatric cancers, and largely because they're understudied and there's been very little progress over the last few decades in there. And then we had to pick different cancers to study. And I chose osteosarcoma because it's a very rare pediatric cancer, which means it's even less studied. And I knew that there was very little known about the genetic origins of it, and so I knew this technology would be a perfect platform to identify new treatment options for osteosarcoma.
I want to be in that clinical trial for soft tissue sarcoma, what would my first step be to come back to the Mayo Clinic?
That's a generally good. Use the mic.
Yes, so we're talking about the malignant peripheral nerve sheath tumor trial?
Soft tissue
Sarcoma. So this study, the way it is designed now is for patients with new diagnosis of soft tissue sarcoma, and we combine the virus with radiation. Again, I can have my contact information available, and you can email me directly. And if that's not the trial for you, maybe we can look into other clinical trials.
Yes.
Will this virus, do you think, replace the chemo?
The question is whether this virus will replace the chemo.
At Mayo with Robinson.
Yes.
who's my doctor. And I don't think, I mean, the chemo didn't really do anything. Was the surgery.
And this is an excellent question. How to best combine viruses? I mean, it might be one day that we'll be so smart about these tumors that we're going to be able to choose one treatment, and this one treatment will be cured. My prediction is that we probably have a long path, and the way to think about it is the virus is not going to replace necessarily the tumor. It will be a different kind of weapon. And we have done a lot of work combining viruses with chemotherapy. And under certain circumstances, because chemotherapy or actually radiation can help the viruses replicate better, it could be one approach to combine them. But I have to say, if I was to compare chemotherapy with some of the immune checkpoint inhibitors, immunotherapy, my prediction, based on what I have seen in preclinical work we have done, is that combinations of viruses with immunotherapy rather than viruses with chemotherapy will be the way to go.
Thank you. Got time for another question? Yes, Harry.
I've got a question for either one of you. Just talk a little bit about the process of getting your work authorized or getting that approval. How long of a road is that? How many roadblocks do you have to face as you go through that to get final FDA approval to actually use it?
I think you'd be good.
That would be good. Yes, I have done that, and I didn't quit, importantly. So there are two different kind of FDA approvals. Is the approval when the drug is first used in the clinic, in a clinical trial, and then it's the regulatory approval when the drug can then be available off the shelf. For patients, you can have your doctor prescribe it. So in terms of the first step, it is a very complicated step. And this is where I'm very thankful for the philanthropy support we have received from different foundations. Because we do have grants from the National Cancer Institute, but frequently these grants are for, let's say, to fund the trial or fund work in mice. This very difficult work, which is optimize your production process or do the toxicology experiments, which they don't really tell you something new. They tell you that the drug is safe. So the NIH reviewers find that boring, but it is critical in order for the drug to be tested in patients. So this is where I think foundations like yours can make a huge difference. I have to say, our motto has been, we're not going to take no as an answer. The reason being that we see patients every day, and I was very pleased to see that the regulatory authorities, after the initial discussion, were willing to see the patient's perspective. It's not the same thing when you develop a drug for sarcoma patients who really need something urgently versus if you develop a new vaccine for healthy infants. Sometimes this distinction is not very clear when you start, at least when we started talking with the regulators. I think I have to say, I feel very fortunate that after 10 years and interacting with them, the approval process has been really streamlined. To give you an example, one of the things we're working on is smarter ways to deliver the viruses. And one of the ways we can do that is use stem cells, because stem cells go in areas of remodeling and inflammation, and tumors are like that. We can infect stem cells with the virus, and then the stem cells go where we want them to go. And actually, we have a clinical trial. We test that in ovarian cancer patients. So I was truly very pleased to see that despite the fact we were proposing a first-in-human study, never done that before, the time from when we send our application, which is called an IND, Investigational New Drug Application, to when we got the go-ahead from the FDA was 1 short month, which means that when FDA has been working with a team for a long time and they trust their work, they're also open to being more flexible. Then, so that's the first step. Then it's the other part which is, okay, what do we need to do for new drugs to really move to regulatory approval faster? And that's, I think, a very, very important and very difficult question. I think the one thing we can do is to encourage patients and participate in clinical trials and physicians to enroll patients in clinical trials. Except if there is a compelling reason. I believe every single sarcoma patient should have the opportunity to be in a clinical trial. And along the lines of what we were discussing before, I feel that our patients are our heroes, because sometimes they're randomized to the inactive drug, but they do it for other patients, for the generations to come. And I don't have a way around it. I mean, we cannot experiment on humans. We need to do it in a way that complies with ethical principles. At the end of the day, that means a randomized study. So encouraging clinical trial participation is, I believe, the way to move drugs fast to regulatory approval.
Want to add anything to that?
That's a good job. I've never gone through the process, so that's
Okay.
new to me.
One last question, yes.
I went to a town hall meeting here last week, my wife and I, in regards to exposure to Agent Orange. I'm a Vietnam veteran. I currently live with smoldering myeloma, and we lost a daughter to leiomyosarcoma back in 2010. The reason I wanted to bring up kind of general information is that it was brought to our attention that with exposure to Agent Orange during the Vietnam era, there were 38 different cancers that are contributed to this toxin. 19 of them are sarcoma cancers. Wondering if the University of Minnesota, Mayo Clinic has any knowledge, if there's any other research going on. And now these toxins, we believe, are being passed on to our children. And that's my exposure to Agent Orange. My daughter dies from sarcoma cancer. So just wondering, just for everybody here, for your knowledge, if you know someone, whether it be a husband or a brother or a son, whoever, that may have been exposed to this, just to check it out. And the VA is looking to compile this information for further research.
Right.
Anything that...
This is a very important question that I believe needs to be answered. We do not have active research at the Mayo Clinic on that right now, but I think in part because the military and the VA is investing a lot on that, and I think it is a very important question to hopefully answer conclusively.
Thank you.
Thank you.